It's an absolute pleasure and honor to be able to introduce Larry Smarr to you, who's giving our talk today. Larry's director of the California Institute for Telecommunications and Information Technology, but behind that lies an incredible career, being a visionary and leader in scientific computing and supercomputing and now data science. I think Larry pulled off some amazing things with the NSF in the early 1980s that certainly I have no idea how to pull off, that resulted in the formation of the National Supercomputing Centers. He founded the National Center for Supercomputing Applications and has been doing amazing innovations ever since. Today he's going to be talking about using supercomputers and data science to reveal your inner microbiome. For the data science students here and listening, and all students listening to this on the streaming and recording too, I really want you to think about what can happen when you take the techniques of data science and really imaginatively apply them into big problems and opportunities that we have today. I also want to say we do have some cookies here, but you might want to have them before Larry's talk because you may not want to eat them after Larry's talk. So with that,
Well, thanks very much. I'm going to talk about a project that involves data science applied to biomedical. The thing that makes it a little unusual is that I decided to use my own body as the data generator. And so we're going to talk a little bit about how that worked. But the main thing is that we are seeing an explosion of genetic data, which for 15 years people have thought of as being human genomic. But actually, as we'll see, the DNA in your microbes is vastly greater in terms of genes, and it's only now beginning to be thought about. So here is 2000 when I moved from Illinois, University of Illinois Urbana-Champaign, to University of California, San Diego. And I started making some changes to improve my health, lose weight, exercise and everything. And I wanted to find a way to keep track of that. So I started off by just getting on the scale every day. And that's one number that, gives you a rough overall sense of yourself. But once I started doing the blood test in 2005, before long I was doing about 100 to 150 variables in my blood every few months. And then I was one of the first people to use 23andMe. So now instead of just 100 numbers over time defining my state, I now had 1 million points along the human DNA, which is where 90% of all human variation occurs, the SNPs. And then I had my whole genome done. So now I'm up to 3 or 6 billion, depending on how you think about it. And then finally with the microbial, we're up to over 1 billion points. And that changes every time you eat something. So if you think about putting the state of your body, I'm a physicist, so that's how I think, into your electronic medical record, you better have a lot of room for the data. And what became clear is that the data sciences is going to have a huge impact on how we think about discovering disease, as I'll show you. So here is a system we use at my institute. This is 32 high-definition screens, each with 2 megapixels, so that's 64 megapixels. So that's 50 times the resolution of pixels that your laptop does. That's something to think about when you get this massive amount of data. How do you see it? And if you're trying to squeeze everything onto your 1 million pixels on your laptop, you're only going to be able to ask certain questions because you can't see the rest of the data. Here, the idea is each of these is a time track of a particular, this is like LDL in your cholesterol, or white blood cell count, or sodium in your blood over, say, 15 years. And you can look at all of them, the whole, I mean, so think of your body as having subsystems, your glucose insulin system, your inflammation system, you know, your heart, your liver, your kidneys, all of these things. And you can just see the whole system working over at once in time. Now, to collect data, I give about 14 vials of blood every quarter and about 6 every month in between, plus stool tests every 2 weeks. And the reason is that your largest immune organ is your colon, among other things it holds the record for in your body. Now, I expected to just see myself getting healthier. And what was very surprising to me was in 2008, I noticed that my inflammation in my blood, as measured by something called complex reactive protein, it's supposed to be less than 1 if you're healthy. And I was up at 5, and then 10, and then 15. And I said, what in the world is going on? And I go to my doctor, and they said, well, how do you feel? And I'd say, fine. And they said, well, why are you here? I said, but I've got data. And he said, yeah, but I'm a doctor. What good is that? And so I knew right there we had a problem. But as you'll notice, I got up to 27 times the upper limit for healthy, which, you know, is not a good thing. And that was in 2012. So I figured, okay, I've got some sort of chronic inflammation. There must be some place in my body that's generating this. And so I started looking at the stool samples because, as I say, the immune system, you're really able to see it closer there. Again, it's supposed to be less than 7, and this is 100, 200, and that's 900. So that's 125 times the upper limit for healthy. And the nice thing is that you can, I don't have biomedical training as such. I'm a physicist and computer scientist, astrophysicist, but I know how to read scientific papers. And so you can go to PubMed and basically teach yourself this stuff. And when you do, you find out that lactoferrin, which is what this is, which is a antimicrobial protein that's shed from the surface of white blood cells called neutrophils, your most common white blood cell, when they're in attack mode, killer cells. That tells you you've got an autoimmune disease, inflammatory bowel disease. I had no idea. So I've got a chronic incurable disease. I didn't know. But thanks to data science, I now do. The reason is I was pre-symptomatic. But after these large spikes in both the inflammation and the immune, then I became actually quite symptomatic. Now, because you're not a medical audience, I'm not going to take you through the gory details of what I discovered. But the main thing is, if your immune system is going that crazy, it's a coupled dynamic system. Think of coupled oscillators. What is it coupled to? Why is it going crazy? And it's your microbiome, because in your body, there are about 10 trillion, there are tens of trillions of human cells, but there are many more microbes. And I'll show you exactly how many in a second. But the other thing, for those of you who are mathematically inclined, you are topologically a torus, like a donut, right? So this is the hole in the donut here, and there's another hole, and there's a GI tube that connects them. And so the surface of your GI is the same as your skin. It's on the outside of your body. So the bugs, even though they seem to be inside of you, are actually outside of you, topologically speaking. And so they're in the environment. And a lot of these things that are autoimmune, the genes in your human genome only account for, say, 20%, and the 80% is the environment. But this is your near environment. So this is why the microbiome is so deeply involved in so many human diseases, and yet today it's entirely outside of medicine as we know it. So the next 5 to 10 years, you're going to see a radical change as we take this dark matter. It's got 300 times as many genes on the DNA in the microbes as in your human cells. So most of the information content in the living cells inside of you is in the microbes. Now, this is, some of you have heard, oh, it's just a myth that there's 10 times as many cells, and this came out in Nature earlier this year. Well, here's all your human cells, but notice that these guys are red blood cells. That's why they call them red. And then platelets. Well, altogether, that's almost all of them, but they don't have DNA in the adult cells. So if you actually talk about the information-bearing cells, then once again, you're back to a 10-to-1 ratio between the microbe cells. And the difference is all of your cells that are human are one species. As we're going to see, these are a vast biodiversity that live inside of you. And in fact, this system, if you think about it as a Darwinian evolutionary system, is about the most intense Darwinian system on Earth. So you know Craig Venter went around the ocean sequencing the microbes in the surface of the ocean, except that there are 1 billion of these microbes per gram in your stool, in your insides. So that's 1,000 times the density of microbes in the ocean. These are incredibly dynamic. This ecology of all these different species working together is constantly changing. So it's very dynamic ecology. And without getting into details, pieces of the genome of one bug can be just horizontally transferred to another one as opposed to dividing in the normal way of reproducing. And this can be quite rampant, actually, this horizontal gene transfer. There are viruses, and those viruses are sometimes called bacteriophages, and they can actually interact very strongly with the microbes. Then your immune system, which is essentially an artificial intelligence system that booted up with no knowledge from birth, by figuring out every time a new microbe shows up in your microbiome, they make an antibody with a memory cell, just like a vaccine works. And that's how you build up your immune system. So the immune system is completely and totally coupled to the microbiome. And if you imagine the microbiome shifting its ecology, then the immune system is going to say, what's going on here, and take action, which is at the heart of why you have autoimmune. But every time you have diets, if you napalm, yourself with antibiotics, you're just blindly killing zillions of these. Most all of these microbes are doing services for you. They are doing vitamin synthesis. They are fermenting the plant polysaccharides that, when they say you should eat vegetables, besides some of the chemicals that are in, it's because they have fiber. And the bugs ferment that fiber, and they produce short-chain fatty acids, which turn out to be the main fuel for the human cells lining your colon. So it's a very symbiotic thing. You just go wipe them out, you're in trouble. And of course, we now know that. And of course, the pharmaceuticals. So the question is, and I'm going to sort of stop with biology at this point and sort of switch into the question, can we use data science to decipher what goes on? And in particular, the way I did this is the NIH spent about $1/3 billion on the human microbiome project. So they said, well, first of all, we don't know what the microbiome is in healthy people. And by the way, it's not just your gut, that's where most of them are, but your oral cavity, your skin, your vagina, wherever, inside of your lungs even, you have these microbiomes. They didn't know what they were. So they got all this genome sequencing going, because each bug is a circular piece of DNA about 4 or 5 million bases long. And so the only way you can tell one bug from the other is to sequence its DNA. I mean, it's really a pure information play. You think of people, oh, well, microbiologists, they put the bugs in little petri dishes and then they put stuff on them and that's how they figure out. Well, yeah, except that 85% of the bugs that are in your gut are anaerobic and therefore die in the presence of oxygen. So you put them in a petri dish, dead, So we don't know what they do. And so the only way we're able to do this now is by looking at their genes. Now, having the genes, that doesn't mean you know their function. So we're in this weird situation where, because of technology, because it's 1 million times cheaper to do this, I mean, this project would have cost me 1 million times as much to do, say, 12 or 15 years ago. So it's that which has made it possible to read out the genomes of these bugs, but we still have this problem of what do they do? Okay, so the neat thing was that after NIH did these 255 healthy people, they put the actual raw reads from the sequencers up as data that you can download, 35 gigabytes or something. So it's a lot of data per person, and then you need 200 or 300 of them, right? And by the way, I could get a few people with inflammatory bowel disease, which comes in 2 flavors, ulcerative colitis and Crohn's. And then I turn out to have colonic Crohn's. Mostly the Crohn's that you hear about that's so debilitating is at the end of the small intestine, which is called the ileum, and that's called ileal Crohn's. And if it's in the colon, it's called colonic Crohn's. Those turn out to be different diseases, genetically in the human as well. And then I had 7 points in time, which as you'll see is greatly increased. Well, that turns out to be 27 billion reads. Now a read is what comes off an Illumina sequencer, and it says, I've got about 100 bases in a row here that I know that that exact order occurred somewhere in this 100 trillion set of bugs over hundreds of species. So think of this as a jigsaw puzzle where you've got, in this case, these are about 100 million of those reads per person. In my case, about 200 million. And you're trying to match those to, say, 10,000 sequenced bacteria or viruses. So that's about 2.7 trillion bases, just to sort of set the scale for the data problem. So naturally, of course, for me, I mean, it is natural. The first thing I think is, let's get a supercomputer. But I mean, you can see from the scale of it that that's the only thing that would make sense. So that's the sequencer at the Venner Institute. This was the Gordon data intensive supercomputer. So Gordon was is the only NSF supercomputer that's been built just for data analysis. I mean, or primarily for data analysis. So that was great. It has big memory, has fast I/O, has all the stuff you want, and has tons of flash memory. And so we downloaded these 3 trillion bases on the healthy people and a few with IBD. Venner actually took my individual stool samples, which are frozen, and then sequenced them. And then we used about 20 CPU years. So, it's like you run your processor for 24/7 for 25 years. And then Dell came in because they got excited. They think bioinformatics is going to be a huge growth area, and in particular data sciences, and it's going to be a growth area. And so we got about 4 million on that. And if you now imagine the output in the dumbest possible way is a spreadsheet, and I've got the rows are the species of bacteria that it could be there. That's about 10,000 rows. And then I've got about 500 columns. And so you end up with millions of, filled spreadsheet cells. And then you set out to do data science. Okay, so not to take you through this in any detail, but you start by getting this big mountain, 100 million of these little pieces of DNA that you've read. And Weizong Li, who was a colleague here of one of you for a while, you have to, of course, because it's coming through you, you will have cells of the human that are sloughed off as well. So you have to first check it against the human genome and remove the reads that actually are from you, not from the microbes. And then you go through a bunch of processes, and I won't take you to the details, to get out the taxonomic profile. So remember, you are in ecology, like an oak forest or a coral reef, right? Or a rainforest, right? You have a zillion different kinds of critters in you. And we want to know essentially a distribution function across all critters. So what's the relative abundance of all these different species? That's the taxonomy, basically. But, as I say, each of these bugs, so a gene in a bacteria is about 1,000 bases. So So in a E. coli, say 5 million bases, there's 5,000 genes. So 4,000 or 5,000 genes, and each gene makes a protein, right? So that's the business end of the bugs. So we actually end up calculating all of the genes, and that gives us then function, because each of those proteins does a certain kind of thing. Okay, so then that's what we ran on the supercomputer for 25 years. Then you got to visualize it. So this is back to the wall, and so each of these, you can see each of these vertical is a bar, and each bar is a species. And this is a log chart of relative abundance, and I've organized them by phyla, which I'll talk about in a second. But for right now, if you just look at it, what I'm doing here is, this is me over 3 different times, say 3 months apart. And over here is a healthy person, a person with ulcerative colitis, and a person with Crohn's. And so I can then compare, and I can walk up to the wall and actually read. You'll see all of these are the names. So let's look at it. We'll look at it a little closer in just a second. Now, let's talk just briefly, because I know most of you are data scientists and not biologists, about the living world on this planet that's been here for 3 or 4 billion years. Almost all of it, almost all the life on this planet, by many orders of magnitude, is single-celled bacteria, archaea, mainly bacteria. And the way that this was figured out by a physicist, I might add, Carl Woese, who was one of my early mentors, is you imagine you got, here's one DNA and here's one DNA, and you compare them to see how different they are. And that gives you a distance metric. And then essentially this thing can show you also essentially the evolutionary history of all life on Earth. And you get this 3-part, this is bacteria, archaea are like bacteria, but a little different. And then here's eukaryotes, and whoops, I must have turned to something else. Oh yeah, there. And let's do that again. Okay. So where are you? There. See the thing says animals? You're at the very tip of that. Now, the difference in your DNA as a vaunted human being versus fungi and slime molds is very small. You look more or less genetically like a slime mold compared to how different the DNA of the bacteria are. And all through you are these different kinds of bacteria. So the distance in terms of biodiversity is huge, huge in the microbiome. And here, by the way, is from 3 weeks ago, was just published, the newest version of this tree. And all of this is bacteria. And down here are the eukaryotes, and down here somewhere is you, right? So this vast world, mostly unknown. In fact, see all those red dots? Those are the ones that are uncultured. We don't know what they do. We just know their DNA. Okay, now I have to stop hitting that. This is not a good design. Okay, so I'm going to just give you a quick way to think about biodiversity. When you think about biodiversity on Earth, you think, okay, fish, snakes, frogs, you know, birds, primates, and so forth, right? But all of these are vertebrate. But it turns out that in the, you know, phylogenetic way we think about life on Earth, vertebra is a subphylum of Chordata. So here, so that's just one phylum. So now, here's a different phylum. That's like all the sponges, that's the jellyfish, that's the radially symmetric animals like the starfish, here's the segmented worms, here's the mollusk, here's all the insects. Each of those is a phyla. And that's about the number of phyla you have inside of you that are just bacteria. So to think of the biodiversity inside you, you got to think of like a zoo and an aquarium and an insectarium, and everything of all those is in you. And the question is, what is the relative abundance of all those species? Well, here's a close-up now of our wall. And again, this is by phyla. So the red and the blue, the Bacteroides and the Firmicutes, are about 90% of a healthy person. So this is an average over 255 healthy people from the Human Microbiome Project. And notice, importantly, it's a relative abundance. That's a log scale vertically. So this bug, which is Bacteroides ovatus, is 10% of all your bugs. And this one, Bacteroides vulgatus, is 7% of all your bugs. So one genus out of all these phyla is 20% of your bugs, of 100 trillion bugs, microbes in you. But it just keeps going like this. Well, straight lines on a semi-log plot means you have a power law fall off, basically. So you have more and more rarer and rarer bugs in you. And so this is the result of the supercomputing output that I showed you. And you think, well, that's great. Why do I care? I mean, surely all I have to worry about is these more general bugs, the ones that are more numerous, right? Well, let's go down, 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 down to say 0.02%. That's called C. diff, Clostridium difficile. That kills about 15 to 20,000 Americans a year, 2/3 of which are in hospitals, because it becomes antibiotic resistant. Why is that? Because you get all these antibiotics because you're getting an operation, you get immunosuppressed, and all of these guys get knocked out. And that lets this weed basically grow and take over the ecological void. And as we'll come back to that in a minute, it's very hard to get rid of it because it's the main thing the ecology is doing is keeping the bad guys down. And if you wiped out the good guys, you know, you try to come in with antibiotics and you have to use stronger and stronger and stronger antibiotics, and that's only about 10 or 20% of the time that it works. And in the meantime, you are miserable sick. Now, here is what I showed you before. That's the 200 most out of 2,000. So this is the 200 most abundant bugs. I'm going to cut it back to just 50. But now we're going to look at 155 different people. And you can see I'm doing it on the wall because it'd be a little difficult otherwise. Remember, I'm having to project 64 million pixels of the wall onto 1 million pixels of this screen, right? So that's why it's hard to see. But the main thing you can get, if you look at the interpersonal variation, the difference between all of you, if we sequenced all of your microbiomes, you'd still say, well, gee, it's mostly red and blue. So it's, you know, mostly these two dominant phyla. But if you look really close, there's something weird about that one. Out of 150, this is 155 different people. That person is 97% proteobacteria. 3/4 of your bugs are E. coli. This is not actually a healthy person. Now, they got into this NIH group because they had a bunch of surveys. But that's just how bad off you can get and still think you're healthy. So think of this as just in terms of usefulness. Remember, this is nothing but data science. There's no wet lab here. Once the thing has been, the wet lab is up to the point of getting the sequencers, but once it comes off as, you know, 100 base units of reads, the 100 bases in this order, everything else is data science. No white lab coats, okay? And so you're able to pick out of 155 people, one person who needs more attention, medical test, right? So from a surveillance point of view, and this is entirely non-invasive, right? You just produce a little stool on a Q-tip and goes in a little vial and gets in the US mail. So, and you can see just how radically different these are. So I'm going to ask 3 questions. Can data science tell the difference between a healthy person and a diseased person without the need of a doctor? We just seen that they can. Let's go look a little closer. And then can you track the time development? And then eventually, can we just look at the genes themselves and understand the functional differences in health and disease? Well, here is the results of this put into just pie charts. The red and the blue, remember, are the two big phyla. In Ileocrones, the blue, which is normally like 3/4 of your bugs, is this little slit right here. It's 50 times reduced. And these, which are called the actinobacteria, which you can hardly see, the little sliver up there, is 100 times larger. But if you take the other form of IBD, which you'd think is very similar, you see these don't look at all like each other. And in fact, the green is the proteobacteria, the E. coli, and it's huge. And I'm sort of a hybrid because colonic Crohn's is sort of halfway between ulcerative colitis, which is confined to the colon, and Ileocrones, which is Crohn's. And so colonic Crohn's is sort of halfway between these two, and sure enough, that's what it comes out. I didn't put that in, that came out of the data science. Now, if you want to do a principal component analysis, you'd say, okay, let's just take all of the blue dots are the 250 healthy people, the purple dots are the Ileocrones, the red dots are me, and for each of these, you have this full ecology. So you have to have something that will take that entire ecology and reduce that to a point in the principal component space, right? And you'd expect, well, they'd just all be kind of jumbled all over each other, right? Not all. So they cluster completely separately. And that has to mean that the ecologies are quite different, which you saw in the pie graphs at the phylum level, right? So here's data science telling you something dramatic about the state of your body when you have this disease. Now, this is one of 80 autoimmune diseases that the NIH recognizes. And you can imagine this for all the others. And it's likely that the microbiome is very intimately involved in almost all the neurodegenerative diseases and almost all the inflammatory diseases, which would include obesity, diabetes, probably cancer, certain kinds of cancer anyway, and heart disease. And yet, you know, we don't have that in medicine today. That's why this is such a fast-growing field. Okay, what about the time development? Well, you know, I mentioned you have to take this whole ecology of all these different bugs and collapse it. What's the way you do that? What is the metric that you take this distribution function and end up with a number? And so Rob Knight, who is one of the top people in microbiome research, joined our faculty in 2000. He developed this thing called Unifrac, and essentially what you do is you remember the phylogenetic tree, the tree of life? Well, put all your bugs on the tree of life, and then look at all the differences between all the pairs of the bugs. And if they're really concentrated, so they're all almost essentially the same, your distance is 0. If they're all laid out over the tree, it's 1. And so this gives you a sense of how biodiverse you are. So now what we're going to look at is a healthy person. Of course, Rob and his wife are the two that volunteered for this, and they did they did oral and skin and stool over a month or 2 months. And I'll just show you the results. So again, this is on principal coordinate analysis axes. You can see that the skin, the vagina, the stool, and the mouth all separate from each other. This is the human microbiome, and this is an earlier version that Costello did. Now, what I'm going to show you is a movie, and each little line is a day. So they took their samples every day, right? And here's what you see. Namely, because they're healthy, essentially, there's oscillation within an equilibrium. So if you imagine a equilibrium solution space to this dynamic system. You're just bouncing around in an equilibrium effectively. You're stable. That's what it means to be healthy, largely. And so as you see, you can actually, there's a lot of space that you could have been in that you weren't. So you stayed, and that's quite a remarkable result given how much difference there is. So Rob, this is Rob and me, this is now, we have taken his code, Emperor, that makes these visualizations from the data and made that run native on the wall. So we can now look at very large versions of this as we get massively more data, as we will. Okay, so that is normal. But what about if you're really sick? Let's go back to C. diff. Remember, you've got a wiped out ecology. Well, this is the miracle cure, fecal transplant. So we take stool from a healthy person and we insert it in your colon using a colonoscope, typically. Those are poopsicles that are. And the amazing thing is that when I first started working with Dr. Sanborn, who is the probably the top clinical researcher in the country in IBD, he was 20 years at the Mayo Clinic, and then he became, we recruited him in 2011 to head up the GI division at our health sciences. I first said to him, look, I've got this problem. Maybe we could do a fecal transplant. He says, oh, we don't do fecal transplants at UC San Diego Health Systems. Okay, that was 2011. He now does 3 a week, personally. This led to a nonprofit, Open Biome, that produces standardized disease-free stool and makes that available to over 500 hospitals in all 50 states for fecal transplants just for C. diff. And that all happened in the last 4 or 5 years. Now, what does it do to you? Well, when you have C. diff and you take that, your, messed up ecology and project it using Unifrac, see these stars? I mean, it's like, that's supposed to be what healthy gut microbiome is like down here, right? They're all up there. So what we're going to do is take this one little star here is the donor, and from that ecology we're going to give the same, from the same donor to all these different patients, and each line is a day. So in basically 1 or 2 days, all those terribly sick people have been converted back to healthy people, and they're now stable. And you think, how can that possibly be? Okay, because you took a healthy ecology of microbes, you put them in, they had all the good guys that can suppress the weeds, they just do their job, and you're back to normal. Now, how many other diseases would this work on? But right now, the FDA does not allow you to get a fecal transplant in this country unless you have C. diff. So there's a clinic like the Taymont Clinic in London. All kinds of people are flying over there to get it for other diseases. They're DIY fecal transplants, which I won't go into much detail about, but I'm not sure, I'm not condoning that. I guess you could say it's the old American spirit of experimentation. But when Rob was setting up his lab, he's gone from coming in January of last year to a white wall lab with nothing in it, to having 25 people, a fully working lab, and he's processed 40,000 genomes as of December. It's from January. It's an amazing guy. And so I felt so excited that I wanted to show how excited I was, so I brought him a welcome present. And these are 40 of my stool samples taken over time and frozen that's on dry ice. And he's now sequenced it. His team has sequenced it. And we're, when we look at it, this is just using the 16S, but we're going to do full metagenomic sequencing. Here's January 2012, 14, and 15. And these are by family of, and you can see there's a lot of variation, but it's not clear what the pattern is. There's not clear anything interesting happens there. Well, what might be interesting? Here's, again, January 12 through July or August of last year. This is my weight. And it's actually, each dot is a weekly average of daily taking the weight. Always exactly the same parameters. And here's 180, 185, 190, and so forth. And what you can see is that during this period, where there's abrupt weight loss, this was while I was on a month of antibiotics, 2 different antibiotics, plus prednisone immunosuppressant. But the red dots are where I have stool samples. Now back then, people thought I was nuts to take my stool and freeze it, you know. I have a very understanding spouse, because we put it in the freezer in the refrigerator until I can take it to school and put it in the proper 80 below zero. Anyway, people thought but you see how bad I was as a scientist? Because I wasn't resolving in time the phenomena. But I started doing it much more frequently. But the thing happened is, once this was over, I started going into this secular decrease in my weight. And it wasn't because I changed my diet or anything. It just, in fact, I couldn't figure out what was going on. And then during this period here, over here, what we've done is taken each of these samples, collapsed it into principal coordinate space. And here were these first 2 off from the edge of everything else. This is when I was most sick, when my CRP was up at 27 and so forth. Then, during this period of weight loss, I was also badly symptomatic. I was having all kinds of problems. I won't go into detail, weekly during that whole time. So then I decided it was time to take some more drugs. So I took, just during this time, 2 drugs, Lialda and Uceris. This is these are both time release. This is an anti-inflammatory, and this is a form of prednisone that's an immunosuppressant. And I was still sick as a dog during the whole time, still losing weight. So that's why after 3 months, I gave up using it. Well, that's the blue dots. And you see now the time evolution here. And over in one side of a cluster. Then, in just 1 month, the weight switched abruptly into this weight gain, and my symptoms began to disappear. And, you know, when this is happening to you, I guess if you were a woman and you didn't understand how her babies come from, and you got pregnant, you'd say, what is going on to my body? You know, this is some mystery. So I sort of felt like that. And the only hypothesis I could have is there must have been a shift in the microbiome ecology, because we know that some microbiome ecologies are very good at making you fat, and others are very good at keeping you skinny. And we do all kinds of mice experiments and so forth to show that, but it's also in humans. Well, you can see that this 1 month transition is just that 1 blue and 1 red dot, and the blue dots are before January 14, and the red dots are afterwards. So I had just after they had done the calculating this, which is gray dots, I said, look, humor me. Why don't we just paint all these dots blue and these dots red and just see if there's anything that jumps out. And of course, there's this complete transition from one cluster to another. And so now what we're doing is digging into the detail of what is different about those 2 states. But it turns out that, in fact, I was right, that there was this massive shift that just happened in 1 month. Now, from out here where doctors live, you know, and I live, I would have no idea that this was going on. You know, and they'd say, oh, well, you know, you're losing weight. Here's a protein shake. Or, oh, you're gaining weight. Here's a, you know, lay off the high glycemic food, you know. Well, I was constant through here with diet. Yeah.
So for that shake, it's just the composition has been changed, or it's just abundance, relative abundance?
So what goes into this is just the relative abundance across the ecology.
But you don't see new species?
Well, so I can show you later. We're digging into that now. And yes, it turns out that if you say, what were the species that disappeared at this point that had been here? And then what are the ones that came into being that weren't here? There's about 84 families, and almost all of them are Firmicutes. Yeah.
So why? What caused the change? You talked about fecal transplants and stuff. What happened?
Well, in a fecal transplant, you have an external change to the system, right? You introduce a bunch of new bugs. Here, the change was I went off of these drugs. And these drugs are directly impacting inflammation. And certain species of bugs love inflammation and flourish in the process of it, presence of inflammation. Other bugs hate inflammation. And so when that happened, I shifted to some other state from the one that these drugs taking every day had kept me in.
So the drugs actually did it?
That's what it looks like.
Yeah.
But that's correlation at this point in time. It's not yet causality. But you can imagine, let's get some mice and let's feed them these drugs and then let's take them off the drugs. And, you know, so what data science is really good at is generating hypotheses. Then you want to go and to do more work to figure out. Okay, final thing. What is the difference in the relative abundance of just the genes themselves? So if you've got 100 trillion cells in the microbes, right, and each one has 5,000 proteins, genes, right, then you've got 500 petagenes in a bag, right? So that's the other way to look at it. And so if you look at healthy people, each of these vertical lines is a person, and this is from that special issue of Nature in 2012 with the human microbiome outcome. And the blue is the blue is the Bacteroides and the green is the Firmicutes, and you can see, again, people are mostly one or the other. But if you look at the genes, if you look at the function, you see that it's pretty much constant across, whereas there's a big change in the taxonomy. And so people say, well, see, you can't just look at the name of the bugs because they're redundant. They have, certain bug can do this and the other bug can do the same thing. So just because you have different bugs doesn't mean you don't have the same function. And so most people think, you know, there's just not much difference in the microbiome. But that's healthy people. So I took my healthy people and I said, okay, let me take one of them, HE11529, so some anonymized person, and we use the KEGG protein families. There's about 10,000 of these. And so we computed for each of these, and this was about 80% of the computing on the supercomputer to do this. That's why you don't have a lot of this in the literature. And I would just take, let's take for each of these protein families, what is the average in across the healthy people? And then for that one person, we're just going to, for each gene, divide the amount that one person has by the average for healthy. That is a sort of a poor way of getting at the interpersonal variation. And you can see that roughly almost all of them are within a factor of 10. And most of them are just 1 or 2 or 3 difference in abundance. But what about a sick person? So here's the elio Crohn's average. Compare that to the healthy. And now what you can see is that for, in this case, 600, 700 gene families, they are 10, 100, 1,000, 10,000 times more overabundant or underabundant compared to healthy. Really? I mean, think what that must mean. You have 1,000 times as many copies of this gene in the disease state than you did in the healthy. No wonder you're sick. But that's not how we think of the disease state, because we're thinking of what the impact of that change is on the rest of our systems, and then that's what we can see from the outside as symptoms. So similar results for the ulcerative colitis and Crohn's. So I'm going to just show you real quickly a couple of screen captures from a much more advanced form of data science called topological data analysis, which a company, IASTI, that was started by the chair of mathematics at Stanford, and they have allowed us to use their software. So we're taking all 10,000 of the kegs times the 60 people. So we have like 60,000 data points that we're using this software on. And the way it works is, if it's red, it's high value. And if it's blue, it's low value. And the color of the rainbow is in between. And effectively, it's making networks, graphs between all of these different data with different lenses. And the lenses are basically the algorithms that you're using for the data science. And what you can see here is that these are high. This is a healthy person. These is also Crohn's disease, and that's ulcerative colitis. And so if you compare, you see this region here is low, and there it's high. Here, this one here, the yellow, these are high and low there. And so you can go around, and when you circle these, they then pull up. These are the lists. This is the the protein family 1134, which does amyl sulfatase. So each one of these has a function, right, as a gene. This is what that gene, what that protein does, right? And so this is very, very powerful data science environment because it's so totally interactive. You just, circle something and boom, these are the things that are causing that. So the reason that's important is because if you've got these cellular networks, and this goes back to a paper 15 years ago by Lee Hood. So imagine that these are all functions that is going on inside of a cell, and they inject this rat with prions, and you see a few of them go to red, and then those begin to propagate through the neighborhood. So what happens is your cellular networks, if they get perturbed in a bad way, that propagates through the thing until finally you're sick. Well, there's formal software tools to do this called Cytoscape, whether it's for RNA or protein expression, metabolic networks, or genetic protein interaction networks. But the trouble is that Cytoscape only works on a megapixel screen. So we've been developing techniques where, say you have 20,000 people, that's the red dots, with 60,000 species, OTUs, and now you're saying, okay, draw a white line from each of these people to all of the bugs they have. That turns out to be about 18 million white lines, and this is on the wall. And it's totally interactive. You can just zoom in and out like that. And so what you're going to want to see is have that network set up, and then have the healthy people, and then have the unhealthy people, and you will just instantly see the differences. And so this visualization of massive data sets is really critical. So I'll just end here by saying, where we're going from this is I now have 50 times instead of just 7. We're probably going to add another couple of hundred to of healthy people that have been sequenced more recently by NIH. We're working with Sanborn, and he's kind of got 300 of his patients that are biobanked with stool and blood, so we can test both their human genome SNPs and their microbiome. And then this is from a Boston study, this risk cohort of ones that have just been diagnosed but have never had any drugs. So you see them early in the disease development. So we got about a $500,000 grant from Illumina of reagents, because what is expensive about gene sequencing is the reagents. It's like your printer. It's the printer ink that costs a lot of money. And then we got, of course, we have now all of, instead of that big diagram of the software, we're converting over to Rob's team, and this is where we're working with Pavel on the assembly piece. And then we just got a 1 million core hour grant on the supercomputer. So again, 800,000 hours is a CPU century. So this is beyond a CPU century. And that's, but even more than that, because the Comet, the new supercomputer at SDSC, is twice as fast as the Gordon cores. It's actually 8 times as much computing time as we did for the previous study. So what we'll be doing over the rest of this year is computing now this much larger number of people, and then doing the data science across all of that. But that'll give us, for the first time, a sort of high-resolution map, both at the taxonomic level of who's there, and then functionally, what are they doing for the genes. So anyway, I'll stop here and we'll have some questions.
So I can send DNA to 23andMe and get it analyzed. Or can I send fecal samples to the data?
Well, commercially, you can send it to Biome, and that will, for under $100, you'll get your microbiome analyzed. Or you can join the American Gut Project, which is a nonprofit that Rob runs, and he has about 7,000 people in it so far. Yeah.
So it seems like it's expensive to get your simulation to run on a supercomputer. Are you able to experiment with smaller versions of the same problem without some computers?
Well, the problem is, with data science, you actually, you have to have enough data that you're covering enough cases that you can really get something that you believe. And when you consider that there's, 5,000 genes per bug, and there's hundreds of bugs, and you need hundreds of people, that gets big quickly. Now, a lot of the algorithm development is done on smaller machines. And then that's optimized both for parallelism and for data transfer and so forth on the bigger machines. But on the other hand, all I had to do was write a proposal to get 1 million core hours. And since I've spent my life, I've run on supercomputers for 40 years. So, it doesn't seem unusual to me. But a lot of the algorithm development is done on smaller machines. Yeah.
Amazing talk. So I see a potential in this technology that can be used potentially by home consumers who are taking many of these medications. So how long do you think, and then getting the recommendation was attention of, well, this is what you're doing right now. It's causing a lot of side effects.
Yeah.
So how long do you think?
I think 5 to 10 years. So fundamentally, here's the deal. Right now, our world, this country lives on pharmaceutical products. And if you ask what the average American, how many pharmaceutical drugs they take every day, and I realize that I am near the home of Eli Lilly, who was a longtime partner of mine at NCSA. So I'm not disparaging the pharmaceutical companies, but fundamentally, the microbiome changes when you eat things. So you can imagine that there are going to be a whole new set of medicinally tuned food, designer probiotics, and prebiotics, which are fiber-based things that the bugs ferment, that you can use to essentially garden your microbiome back to health. Now imagine that instead of 7, we have 100,000 people who are in the cloud with their microbiome, and we have all of their symptoms and how healthy they are and how much they weigh and how old they are and all that. So yeah, you just take a little sample, put it in the vial, goes up in the cloud, And your microbiome is then compared to all of these other microbiomes, and you're a point in 100,000 or a million or whatever people, and people with microbiomes like you look like this, have these problems. Then, if you want to go from there to some other microbiome, here's a set of procedures, which might include eating fermented food, which has live cultures in it, you know, changing essentially the environment, the pH, other things that the bugs are sensitive to. They're sensitive to, sometimes they need iron, sometimes they need other things and so forth. But effectively, getting away from this, putting a pill in your mouth that disrupts a lot of your systems, and it does not take into account at all what it's doing to the microbiome. It's very point sort, you know. So I think it's going to be kind of a philosophical change, and of course all the major pharmaceutical companies get this, and so they're working a lot on the microbiome. Bye-bye. He and I knew each other back in the day. Yeah.
One of the things, like you mentioned about like taking the samples and freezing them and all of that, right? So like we keep hearing about the dissolvable chips in the brain to measure the concussion and things like that. Is there anything besides this area? How do you instrument your body in some ways to give out this data without sampling or so on?
Right. Well, if you're trying to do values of various chemicals in your blood, there's various techniques that are underway. So, and one thing in particular is just non-invasive glucose monitoring. That would make a huge impact if we could do that. But so far we can't. You have to put a needle in, do a blood prick, something like that. On the other hand, your hand is transparent in infrared, and you can see the veins, and you can, if you can look closely enough microscopically, you can see essentially the shape distribution of molecules. So there are various people trying various things to get non-invasively to get this sort of thing. Given that you're sequencing it, you still need to get it into a sequencer, which are these big machines, right? But there's a new device which I haven't tried yet, but I'd like to. So it's like a big pill, and you swallow it, and it goes all the way down through you and out the other end. And along the way, ever so often, it takes a little sample. And you think, well, gosh, how can a pill do that? Well, given that there's 1 billion of these bugs per gram, if you get a milligram, you still got 1 million bugs, right? So by taking tiny little bits along the way, and in particular in the small intestine, that's a very difficult place to get to, and yet a lot of diseases are located there because of imbalances in the microbiome. So that's an example. Then when it comes out, you take it and you then sequence all those, and you essentially get a geo map, sort of a map of the change of the microbiome. It does change a great deal along your GI tract from your mouth to the other end. So, but right now, our ignorance is so great about the state of things that just doing it the old-fashioned way is giving us, you know, tremendous amount of insight. Okay, David.